been a long time since we made a video and we apologize for that beforehand. Hope you guys understand. With that out of the way, let us delve deeper into the realm of carcinogenesis and try to understand what exactly cancer cells do differently as compared to non-neoplastic cells. However, before we do that, it is crucial for us to go over the various pathways that are involved in a normal cell, as the receptors, signaling proteins, and the transcription factors involved in them are the major site for various mutations, translocations, and even amplifications. As we studied previously, we had the various pathways that a normal cell uses. Among them was the PI3-AKT-mTOR pathway. We had the beta-catenin pathway. We had the RAS, RAF, MEK, ERK pathway. And we have the NOT signaling, right? And we have the Janus kinase pathway. And the TGF-beta signaling pathway, right? So these were among the various pathways, and in this video we will have a discussion on the RAS, RAF, MEK, ERK pathway. So what exactly is RAS? RAS was the first oncovirus that was implicated in causing cancer, and RAS superfamily of proteins were defined in the rat sarcoma viruses, and hence the name RAS, that is rat sarcoma virus. So what are these RAS proteins? So well, they are small GTPases, that is G proteins with a monomeric subunit, unlike the unconventional trimeric G proteins we see in GPCR signaling. Like the G protein coupled receptors and the G proteins, they're very similar in action, but we only have a single subunit, that is they're monomeric instead of trimeric. RAS also operates as a biphasic switch, that it is either on or it is off. When it is in the on state, it is often associated with GTP. When it is in the off state, it is often associated with GDP, right? And as we know, RAS are ultimately GTPases, and hence they will convert the associated GTP and will get inactivated as they will form GDP. And this process is entirely intrinsic. However, we have some proteins that actually influence this state of the on and off state, as we will discuss further. These proteins, that is the RAS proteins, they're often present on the plasma membrane, and they're bound to the plasma membrane by a 15-carbon farnesyl group. How does this protein get activated in the first place? Does it get influenced by extracellular mitogens acting via some other unknown receptors? The answer is a definite yes. The answer to this puzzle came with the identification of a rather peculiar receptor in Drosophila. Now let's just turn our attention towards that. The genetics of eye development in the fruit fly Drosophila melanogaster revealed a series of genes whose products were essential for the normal development of the ommatidia. Now, what are these ommatidia? They are light sensing units in the Drosophila fly. Now, one important gene came here to be known as the sevenless. That is, in its absence, the seventh cell in the ommatidium failed to form, and that is why the term sevenless. So, provocatively, after cloning and sequencing, the sevenless gene was found to encode a protein with the general structural features of a tyrosine kinase receptors, which we have already discussed. Specifically, it was supposed to be a mammalian FGF receptor. The first clue to the puzzle was the identification of sevenless. Now, the sevenless is a homologue of the FGF receptor in mammalian cells, as we studied. Now, the ligand, that is a growth factor ligand, will often bind to the sevenless receptor, and it is often associated and the ultimate activation of RAS protein. But what came in between? Now we need to focus our attention on that. Other proteins were isolated that transduced the signals from the receptors, and one of these downstream proteins was encoded by a gene that was named the son of sevenless, as it came just after sevenless. There is vertical, you can call it, like in the lineage, so sevenless, and it is often associated with the son of sevenless, and that was transducing the signal to RAS. We have got to study what exactly is son of sevenless. So son of sevenless, or simply SOS protein, is a GEF protein, or it is a guanine nucleotide exchange factor protein. Now, what is its function? That it replaces GDP associated with RAS with GTP. That is, there is exit of GTP and there is association of GTP. And what are these factors? These are known as GEFs, that is guanidine nucleotide exchange factors. On the contrary, we have some factors that actually catalyze the breakdown of GTP into GTP. That is, they activate the GTPase domain of the RAS protein, and these proteins that actually activate the GTPase domain are known as GAPs, that is GTPase activating proteins. And one among them was neurofibromin 1 gene that we will consider in another video when we will finally study about the tumor suppressor genes. So other intermediates in this cascade were soon discovered, like GRB2 and the SHC, that is Src Homology proteins, were discovered that were soon intermediates in it. One thing that we need to discuss is some domains, like son of sevenless was associated with Ras. What is this interaction, how it is mediated by that? Or we say ki GRB2, the GRB2 protein is associated with son of sevenless. How is that mediated? It is mediated through some motifs, it is mediated through some domains. Now we have the primary domains that we need to study is the SH2 and the SH3 domains. They stand for Src Homology 2 and Src Homology 3 domains. Now what are Src Homology 2 domains? They are basically, the ligand for them is phosphotyrosine. And we very well know that if there are protein phosphotyrosine residues over here, then GRB2 will associate with these phosphotyrosine residues through Src Homology 2 domain, right? Now there is another domain known as the Src Homology 3 domain that often associates with proline-rich regions in other proteins, which we will study once we go into the details of the Ras, RAF, MEK, ERK signaling pathway. There is a protein known as SHP1. Now, what is this protein? It is actually a phosphotyrosine phosphatase. That is, that if this is the associated receptor, it will cleave the phosphotyrosine domains that is present due to the autophosphorylation of the tyrosine kinase domains that are present over here. That is, when this domain gets dimerizes with this domain and there is activation of phosphotyrosine, an ultimate conversion of tyrosine residues into phosphotyrosine residues, it will cleave the phosphate and basically exerts a negative feedback inhibition over the excessive signaling, right? Coming back, we have SHP domain, that is a phosphotyrosine phosphatase domain. So why are we exactly studying this? There is a very interesting fact regarding this SHP1 protein, that is often the EPO, that is erythropoietin receptor, it associates with the EPO receptor, that is a tyrosine kinase domain. Now we know what is going to happen. Now, if we assume this is the EPO receptor and the ligand binds, that is EPO, then it will phosphorylate these residues over here. The story is fine. Now, we have SHP1 domain. After the job of the EPO is done, it will cleave off the excessive phosphate residues on the tyrosine kinase domain. Now, what if this goes wrong? So, there are some individuals that inherit a mutant gene encoding an EPO receptor that lacks the usual docking site for SHP1. What does this mean? That this protein, that is the EPO receptor, it often lacks the domain for SHP1. Now what will happen? SHP1 is not able to bind to the EPO receptor. Now that will lead to excessive stimulation, that is the excessive secondary messenger system that will transduce to the EPO receptor. Now what will happen? So we have an hyperactive EPO receptor. What will that lead to? That will lead to increased RBCs and that will lead to increased oxygen carrying capacity. Now, there was this one Finnish family that won Olympic medals back to back, that it ran in the family that they will definitely win the Olympic medals. Now, this was associated with a mutant EPO receptor that didn't have a docking site for SHP1. Now, that's interesting.